last-minute surprising guest, who's Meredith Schuman, who is going to talk about the how. Go ahead. She's going to talk about how remote sensing can contribute to the characterization of the genetic diversity of the European beach across its entire range. So we are happy you came from Switzerland for both fieldwork and your contribution to this meeting. So thank you very much.
Merci beaucoup. Thank you very much. Yeah, I'm very, very happy that it worked out. Thank you. And I'm trying to give you just a quick taste of what we're doing in my group. And I'm very grateful to Elodie and Joseph and to the Friends of Massane, because this work that I'll be telling you about, part of it started in 2019 in Massane, actually with my daughter when she was, I believe, 4 or 5 months old, and now she's here 4.5. So it's really time that we build on it together. And we're looking forward to it. So what we do, I'm leading a group called Spatial Genetics at the Univ.Zurich, and we're interested in using ways to do high throughput assessment of genetic variation on the scale of landscapes to species ranges. And one of the species that we've become very interested in working with is European beech, for many reasons that I suppose I don't need to convince most of you of. For our group in particular, one reason is that there's an excellent database of remote sensing data for beech that's relatively well annotated, at least in some forests, and the image I show here is unfortunately not Massane. It would be really wonderful to have an image like this, and for the reserve area, I think we actually have the data that we would need to do it. This is the Lagon Mountain, which is close to Zurich at the border between the cantons of Zurich and Aargau in Switzerland. And what you see portrayed here are on the one hand a three-dimensional model of the trees in the forest, and Felix Morsdorf, who generated this model, is actually not happy and says that meanwhile, of course, we can do much better, because you see the trees are kind of, they look a little bit chunky. It's kind of like looking at little pieces of a cliff face, and of course we can do a much more realistic modeling of trees, but the reason that the trees look like this in this model is that the data to make that model came only from above, only from aerial laser scanning. And if you combine aerial and terrestrial laser scanning, you can get a very nice, pretty precise three-dimensional model of individual trees, even on a larger scale. And then the different colors you see here are estimates of physiological diversity as measured in terms of relative contents of chlorophyll, carotenoids, and water, as estimated from aerial imaging spectroscopy. So basically estimating this by typical absorption features in the spectrum of light or electromagnetic radiation. And so the areas that are blue are more similar in these traits, and the areas that are yellow are more dissimilar in these traits. Generally, we're working in this larger context and contribute to a growing number of groups who are interested in combining data gathered on the ground, which is what I'm more familiar with from my background, really crawling around on the ground and collecting samples and ecological data, up to satellite-based sensors in order to help get the big picture of biodiversity from smaller to larger scales. So we want to know why plants are different. We're focused on plants in the spatial genetics group because I'm a plant person, where it matters for ecology and for people, and we use many different approaches to get at that question, as I mentioned, from doing extractions and analyses in the lab of different natural products that plants produce, as well as their nucleic acids, to high throughput sampling of chemistry in the fields, and also to different kinds of chemical and optical remote sensing. So I want to mention why we're particularly interested in European beech, although, like I said, I don't think I have to convince you that it's an interesting species to study. So of course, as you know, it's a keystone forest species across Europe. And actually, I was really excited to see a very similar picture in Isabel's talk as the one that's up here in the top left. So it's recently transitioned to higher concern, in part exactly because of such observations that in beech forests across many parts of its range, in 2019, we've been able to observe, or this observation comes from 2019, people have observed that there are beech that are clearly suffering, apparently from recent increased intensity of droughts, especially the 2018 drought across Europe, right next to beech trees that seem to still be growing well and are healthy. And so there's also been a study done by a German group that looked at a few populations in the southwestern part of Germany to ask whether this could be explained by differences in genetic markers. And we also recently did a study that we'll be submitting as a preprint soon in collaboration with a group that does monitoring of beach across Switzerland. And I think it's very exciting to see more of these studies done because, for example, what came out of our analysis was that the answer to this, what's the genetic structure and are there genetic differences, was different depending on the site that we looked at. And also in the locations on the genome that we found that did differentiate these trees at many sites, we had no overlap with what was found by the German group, even though we were using similar methods and the same reference genome. So this seems to be quite specific to different sites for many reasons that I would be very happy to talk about, but can't go into in this talk. So anyway, an aside. Yep, and so when I started working in this group, the first annotated beach genome had just been published. And so along with the fact that there are meanwhile decades of population studies, and you already from Hans nicely showed probably the most complete study from Magri et al. In 2006. And we, in 2018, 2019, and with literature suggestions from Elodie, wound up doing a systematic analysis to put together all of the population genetic work that we could find that's ever been done on European beach. And so that's what this map represents. The different colors are different types of molecular markers, and the different circle sizes are different numbers of samples. And this is limited to the geolocated samples and comes from a total of about 50 years of work. And then we also tried to update what we know about genetic diversity across the range by comparing studies that had been done with different sets of microsatellite markers. And if you're interested in that, this was led by Camilla Stefanini, a novel synthesis of 2 decades of microsatellite studies on European beach. Anyway, so there are lots of reasons that we're interested in beach, also because of this natural hybrid zone with oriental beach, which expands the genetic diversity across the species range that we could potentially access using remote sensing. So I want to give you a couple of examples of how we've done this, and then focus on a couple of data sets from Massane, and then I'm sure I'm already done in terms of my time. So one recent study that came out of the group, and this has been in close collaboration with the remote sensing group at the Univ.Zurich, led by Michael Scottman, was led by a PhD student, Eva Chiss, who actually also came to Massane in 2019. And she used this Lagoon forest that I showed you at the beginning, and just a very small area in the Lagoon forest where we have a nice one-to-one crown map of the dominant beech trees. So in this little patch of the forest, we can align imaging spectroscopy data that we collect by flying over in an airplane to individual tree crowns. And that's been achieved using a combination of terrestrial and aerial laser scanning with aerial imaging spectroscopy, like this image I showed you at the beginning of the talk. So anyway, Eva was able to summarize about 10 years worth of imaging spectroscopy data from flying an airplane with an instrument over this forest and measuring the electromagnetic radiation from the sun that was reflected back from the tree canopies. And when you do that, you get an average spectrum that looks like this. So this is the white line. The x-axis here is wavelengths of radiation from 400 nanometers up to 2400 nanometers. So this is the visible range here from about 400 to about 700, 750 nanometers. And then this part here is in the infrared outside of the visible range. And so for those of you who aren't so familiar with this kind of data, plants generally absorb a large part of the visible spectrum, right? They use it for photosynthesis. And then outside of the visible spectrum, we have the so-called red edge, where we get much more reflectance from plant canopies and leaves. And from these data, we can estimate things like pigment content, water content, and aspects of leaf and canopy structure, and also relative abundance of non-pigment materials that are present in relatively large amounts, like condensed tannins and phenolics, and structural carbohydrates, and this kind of thing. So anyway, Eva took about 10 years of imaging spectroscopy data over the site, and she also had microsatellite data from 68 dominant Fagus sylvatica trees in this patch. And so on the one hand, she used Ney's genetic distance to assess the distance among the genetic distance among these trees in a matrix. And on the other hand, she used the cumulative spectral data from 10 years to ask how far apart are these trees from each other in spectral space, and how much does that change? And what she wanted to know was how well does the distance in terms of their spectral reflectance correlate to their genetic distance, and how does that change over time? And so what's shown here looks quite complex because you have all of the abbreviations, but you might recognize some of these. So the abbreviations on the y-axis are all different climatic variables that she was able to obtain for the site over the time of the observations. And so what she wanted to know was, particularly to the extent that there is any association, does it get stronger or weaker in relation to different climatic conditions? And what she found in this case was, with this data set, that she could get up to about 40% correlation between spectral distance and genetic distance as calculated by these microsatellite markers, and that this was achieved particularly with high cumulative growing vapor pressure degree days, so relatively dry conditions, and also with a large number for cumulative growing degree days, so relatively hotter conditions. And this was also true when she looked at temperature. I think this was the temperature up to 11 days before the acquisition as a variable. So generally under hotter and drier conditions, she was able in some parts of the spectrum to get up to a 30 to 40% correlation between the spectral distance and the genetic distance of the trees, and these relationships were much weaker under other conditions. So this is what came out of the study. It's very interesting, and she also tried to estimate the uncertainty, so that's what this is here. The very dark parts are parts where we know that just from our ability to retrieve this reflectance data flying over the canopies, we have very high uncertainty. So for example, this red bar here that has a nice correlation is also in a zone of very high uncertainty, so this would not be such a good bet. But there are other parts here around this red edge, for example, where we have relatively high predictive power and relatively low uncertainty. And something that's interesting that I just want to mention that may also be interesting to you, in human genetics just this year, it was recently published that microsatellites are not neutral, but they actually bind proteins to regulate gene expression. And I'm very interested in whether this happens in plants as well, because it would make this result make a lot more sense. Happy to talk about that. Okay, so can we remotely sense genetic variation? This is one proof of principle that we can get some relevant information out of spectroscopy data. In order to do this, I also wanted to show you this because this is actually relevant for data sets that also exist over Massane. So in 2021, as part of a campaign with Avris NG and NASA's Jet Propulsion Laboratory, this is their flagship imaging spectrometry instrument. We flew over many beach sites around Europe, including La Massane, with support of Elodie and Joseph. And we obtained data where the projection in red, green, blue space looks like this. So we've got you can see flight lines over an area. This is also unfortunately not Massane, because for this test data processing, we used something smaller and less complex. This is a site in Switzerland where we're interested in this little patch here. This is the quality in terms of resolution that one gets normally from these data, and the data at this resolution can already be downloaded from the NASA JPL website. The full data also, not just these RGB projections in visible light. However, with the remote sensing group also, and particularly Kimberly Mason, we've been working on an improved processing pipeline that allows us to get much higher resolution data. And this is actually one of the steps that we want to have to be able to do a good job of assessing genetic variation so that we can really distinguish individual trees. Because you see, even with this much higher resolution, we've got enough problems with light and shadow and other things to deal with. So it's a much better starting point. So hopefully by January, February next year, Kimberly will have been able to reprocess all the data, and also these high resolution images will be available over La Massane. Okay, and then also back when we came here in 2019, we did terrestrial laser scanning for the reserve area of La Massane, and this we've also passed on to Elodie and Joseph, and I just wanted to show you what the results of that were after the data were assembled. This was done by Felix Morsdorf. The little lighted up areas are areas where we were able to automatically detect individual tree stems, and the size indicates cylinder fitting, so it gives us some estimate of tree size, or DBH, and this is a digital elevation model that was generated also from these LiDAR data. And as you see, you can get a much better model of the individual trees than you saw on the first slide. Okay, so finally to conclude, I wanted to show you that we have done some leaf spectroscopy in Massane. So we took leaves from individual trees, and we did exactly the same kinds of measurements, but not from an airplane, but with direct contact on the leaf, which is what Julia and I want to do starting tomorrow again. And this was an analysis led by Fanny Petitbon, and she was interested in how much variation is there within individual trees in their branches, between trees at one site, which was La Massane, and between trees across sites, which are some sites in Switzerland. And we were happy that she was able to show, this is showing relative variation in the data. So it kind of looks like an inverted spectrum, that trees within forest sites are more variable from each other, so that's the orange bit, especially in some parts of the spectrum, than leaves within a single branch of a tree. And also across sites, there's a bit more variability than there is within one single site. So this was an important building block for us also to say that we should be able to detect relative genetic variation using spectroscopy data, right? If we can see bigger differences between trees that should be more distantly related. But a lot of assumptions go into this, so it was only one proof of principle. Meanwhile, we're working on a common garden of beech seedlings from sites across the range, including La Massane, which is this FRM site here, and also with a library of some combination of leaf and aerial imaging spectroscopy data, whereby we certainly have the best data and annotations for the Legan forest and for Massane. And I also wanted to quickly show you that we use some genomic data from Massane, but from only 10 trees as part of a relative analysis that Eva, the student who did the aerial study, also did across the range. So there are issues with this analysis, but it also, I think, is consistent with what you've already heard about what we already know and expect about the special nature of the trees in the Massane forest. Good. And here Eva was trying to do then a prediction of in a different way, how well we can predict genetic diversity from spectral variation. And this I'm happy to talk about in more detail if anybody is interested. But what she could conclude was also taking a completely different approach, that there does seem to be some genomic information in the spectral data, but this has, I think, a long way to go. So with that, I'd like to thank you very much. Joseph, I never manage to correctly spell your name, and I am so sorry, but I'm very grateful. And I'd like to thank the group, and particularly highlight Eva, Fanny, and Kimberly, who did the work that I showed you. So thanks very much.
I'm afraid we're a little bit short on time because it's lunch in 4 minutes. So you're all invited for lunch, by the way, at the restaurant of the observatory. So maybe just one quick question. Otherwise, we have time to discuss with Meredith during lunchtime.
I've got a question. Thank you, Meredith. It was a really nice talk, very impressive results. I was wondering, with this huge amount of data, how do you deal with it? Do you use now like artificial intelligence to help you, or because it's really huge? Thank you.
Yeah, thank you. That's a very good question. So I want to start in kind of a stepwise manner using more sophisticated models. What we've used up to this point for the prediction of genetic variation has been either simple correlation, so what's the correlation coefficient Or in Ava's case, she was first performing RDA and then again simply doing correlations. And so I think that there's much more information we can get out of it than that. In the beach system a bit, and also in another system, we've started to do genome-wide association studies with spectroscopy data. So this is, again, we're using well-established linear models. And the contrast between, for example, the heritability estimates that we get out of that as opposed to the gene-by-gene associations that we're able to find is already very informative. Where I think we need to move forward is definitely to leverage deep learning to enhance the image analysis that we're doing with spectral data and to enhance our ability to find nonlinear associations and do classification well, which is what it's good for. And I think we also already, as a next step, will have something to learn with some fairly simple modifications to GWAS models that allow for epistatic interactions.
Thank you.
Thank you. Merci. On se retrouve au restaurant de l'Observatoire pour le déjeuner. Vous êtes tous conviés. Et rendez-vous ici 14h10 pour reprendre à l'heure 14h15. Merci. Thank you.